Assalamualaikum and hi everyone. My name is Muhammad Haikat bin Fauzi and my metric number is 22302280. My group will discuss about Salmonella enterica and I will share about the importance of understanding host-parasite interaction, virulence and pathogenicity, and also the overview of Salmonella enterica. Importance of understanding how pathogens cause disease, how hosts react, and how host infections can be prevented or controlled. It is essential to understand host-parasite interaction, virulence, and pathogenicity. The development of specific treatment, vaccination, and public health efforts to reduce the worldwide burden of infectious disease is guided by this understanding of the process pathogens use to reach, remain, and harm the host. Salmonella is a genus of gram-negative bacteria that cause a variety of foodborne diseases in humans. Millions of infections are caused by these bacteria each year, making them a serious global public health risk. Variants of Salmonella enterica, including Typhimurium and Enteritis, cause non-typhoidal salmonellosis, which is characterized by fever, cramp in abdomen, and diarrhea. Typhoid fever and a serious systemic infection that is common in areas with a poor hygiene is carried by Serover typhi. Salmonella is virulent because of its capacity to enter host cell, avoid immunological response, and generate virulent agents such as endotoxins. Because of this method, the bacterium can cause systemic infection in extreme cases or localized infection in the digestive system. To counter Salmonella's impact on world health, it is essential to understand the pathogenicity in order to develop and improve digestive equipment, antimicrobial treatment, and also vaccine. That's all from my part. Thank you.
My name is Alifaika bin Ayub and my metric number is 22301823. I will continue to describe about host parasite mechanism that was described by my friend Ahmad Wafi Hadi just now. Type 3 secretion system 2 T3SS2 will be encoded by Salmonella pathogenicity island 2 SP2, which secrete effectors like SIFA, SSEJ, and SSEF. These effectors will remodel the CSB membrane to stabilize the structure while also inhibiting lysosomal fusion and preventing bacterial degradation. Furthermore, SIFA effector play an important role in maintaining vacuole integrity, while SSAJ will contribute to lipid modification of the vacuole membrane so that it becomes more suitable and favorable for the bacterial replication. Other than that, Salmonella can secrete effector proteins called AVRA. AVRA can suppress host inflammatory pathways by inhibiting NFKB activation. NFKB is an important protein for cytokine production. Moreover, Salmonella can evade autophagy by producing effector called SOPB. Autophagy is a mechanism used by hosts to degrade the intercellular pathogen. The SOPB will interfere with autophagosome formation, thus inhibiting Salmonella degradation.
Hi, my name is Ahmad Wafiadi bin Muhammad Roslan, number matrix 22303078. I'm going to explain about host-parasite interaction of our microbes. Salmonella enterica use a series of mechanisms during the host-parasite interaction. The key mechanism of host-parasite interaction depend on type 3 secretion system. The first mechanism is host cell adhesion. It's about how Salmonella colonize the intestinal epithelial cells. It uses membrane and adhesin, which is a specialized surface structure on Salmonella. The bacteria use type 1 membrane to bind to mannose-containing receptor on epithelial cells. adhesins such as SHDA and SFA will further target extracellular matrix protein on hosts like fibronectin and laminin to secure attachment. Another fimbria, called SAF fimbria, involved in biofilm formation. Biofilm will protect the bacteria from environmental stress. The next step is invasion and intracellular survival mechanism. It's about how Salmonella invade the host cells and establish a niche within the host intracellular environment. Salmonella use type 3 secretion system 1 that are encoded by Salmonella pathogenicity island 1 to inject effector protein such as SOPE, SOPB, and SPTB. SOPE will induce actin polymerization, causing membrane ruffling and creating a cup-like structure around the Salmonella, which forming a phagosome. While SOPB modify the host signaling pathway, which promote bacterial entry. SPTB, however, are used to regulate SOPE and SOPB. In phagosomes, the bacteria resides inside the Salmonella-containing vacuole, which establish a niche for replication of Salmonella.
The pathogenic process of Salmonella enterica can cause inflammation or disease in animals' digestive system. This process consists of several steps that enable Salmonella enterica to invade host cells. First, to approach the intestinal epithelium of the animal, motility and chemotaxis are required to establish initial contact with the host cells. Motility is crucial to increase the chances of attachment, allowing the bacteria to continue the pathogenic process. The pathogenic process of Salmonella enterica can cause inflammation or disease in animals' digestive system. This process consists of several steps that enable Salmonella enterica to invade host cells. First, to approach the intestinal epithelium of the animal, motility and chemotaxis are required to establish initial contact with the host cells. Motility is crucial to increase the chance of attachment, allowing the bacteria to continue the pathogenic process. Next, the attachment of the bacteria to the host cells relies on fimbriae and adhesins. Salmonella use fimbriae, which are hair-like structures, and adhesins to stick to the host cells. There are several types of fimbriae, including type 1 fimbriae, curly fimbriae, PEF fimbriae, and STD fimbriae. The most crucial type is PEF fimbriae, as it helps bacteria attach to a specific receptor called the LEX antigen on the host cells. Adhesins also assist Salmonella in attachment and colonizing the intestine with protein such as SIEE and BAPA, playing significant roles. After attachment, T3SS1, a needle-like structure, injects DNA into the host cells. This DNA induces cytoskeletal rearrangements such as ruffling of the host cell's membrane. When the membrane is ruffled, it allows the engulfment of the bacteria. The injected DNA triggers inflammation, leading to disease of the animal digestive system. Conclusion: this is the pathogenic process by which Salmonella enterica invades the animal's digestive system.
Assalamualaikum and hi everyone. My name is Nuri Izzah Zahirah binti Muhammad Zamri, metric number 22301181. For my part, I will talk about the virulence factors produced by Salmonella enterica. Virulent factors is a molecule or substance produced by bacteria that enable them to cause disease in hosts by evading the host defense. The first virulent factors that we're going to talk about is type 3 secretion system, T3SS. T3SS function for translocation of proteins from bacterial cytoplasm into host cell. T3SS have two types, which is T3SS1 that encoded by Salmonella pathogenicity island 1, which necessary for invasion of host epithelial cells. It induces cytoskeletal rearrangement that promote bacterial uptake. Another type is T3SS2, encoded by Salmonella pathogenicity island 2. It facilitates intracellular replication and survival. Next virulent factors is adhesins. It is essential for colonization host tissue that include fimbriae and outer membrane protein. Fimbriae attach to intestinal epithelial cell and mucosal surface, while outer membrane proteins enhance bacterial binding for a successful invasion. Salmonella enterica's outer membrane contains lipopolysaccharide, which is one of the virulent factors. It serves as a physical barrier, shielding the bacterium from antimicrobial peptides and complement-mediated lysis. Toll-like receptor 4 on host immune system also recognize lipopolysaccharide, which set off inflammatory reaction. Next is flagella. Flagella playing a key role in the bacterial motility and ability to infect host tissue. It increases motility and chemotaxis, enables movement through the intestinal lumen, and assists in finding favorable colonization sites. The last virulent factor that we'll talk about is VI antigen, which is a polysaccharide capsule. VI antigen shield the bacterium from host immune response and inhibit complement activation and phagocytosis.
Assalamualaikum and greetings. My name is Muhammad Azam bin Abdullah. Number matric 22303088. Pathogenicity will explain how Salmonella enterica can cause disease. The main disease that is caused by this bacteria is salmonellosis, a type of gastroenteritis or abdominal inflammation. Salmonellosis will cause symptoms like diarrhea, abdominal cramps, fever, and vomiting. The way this happens is when S. enterica enter and invade the intestine's epithelial cells, causing disruptions of its normal function. Its mechanism involves the most important virulence factor, the type 3 secretion system, or T3SS, that inject bacterial effector proteins into the host cells to manipulate the host cell to help bacteria proliferation. The mechanism starts with ingestion of contaminated food or drinks. Then, the bacteria must survive stomach's acidic environment by activating its acid tolerance response. Once it adapted to the low pH of the stomach, the bacteria will pass through the stomach and attaches to M cells in Peyer's patches in the intestinal epithelium. Invasion of bacteria starts going into enterocytes or follicle-associated epithelium by using the T3SS virulence factor. Our immune system will then retaliate by sending phagocytes like neutrophils, inflammatory monocytes, and dendritic cells to engulf and destroy the bacteria. However, the bacteria that still remains in the intestine will start to produce toxins that damage the intestine tissue and causes diarrhea. Then the bacteria stimulates the release of pro-inflammatory cytokines that causes inflammatory reaction, causing severe abdominal pain. The bacteria can also cause systemic dissemination, which can spread them to other parts of body like liver, spleen, and bone marrow that can cause further complications.
Greetings, I'm Timingi and I'm going to continue the presentation of the pathogenicity of Salmonella enterica. Another common disease caused by Salmonella infection is the typhoid or paratyphoid fever, and they are both caused by Salmonella enterica serotypes typhi or paratyphi A, B, and C respectively. Typhoid fever can be life-threatening if lack of appropriate antibiotic treatment, and after infection, the symptoms include stepwise fever, abdominal pain, diarrhea, and also raw spots. The pathogenesis mechanism of typhoid fever is generally similar to the one of the gastroenteritis. The infection of Salmonella typhi is through fecal oral route, which is by ingestion of the contaminated food and drinks. The acid tolerance response allows Salmonella typhi to survive the gastric acid and enters duodenums, finally penetrate through the epithelium and enter lymphoid system. Which, with type 3 secretion system, the multi-channel proteins allow Salmonella typhi to inject its effectors across the intestinal epithelial cell membrane into the cytoplasms. These effectors triggers the reconstruction of the actin cytoskeleton of the host cell, causing outward extension of epithelial cell membrane and engulf the Salmonella. The effectors also works to reconstruct the vacuole surrounding the Salmonella after being engulfed into the host cell. This vacuole resists the fusion with lysosome inside the host cell and prevent the digestion by the lysozymes. This allows Salmonella to survive within the macrophages and the host cell to reproduce. And this is all from me. Thank you.
Hi, my name is Muhammad Mukris Hakim bin Muhammad Sham with metric number 22302982. Today, I will present about the clinical importance of the Salmonella enterica. Hi, to begin with, Salmonella enterica is a common bacterium that can cause food poisoning. It is found in the intestines of many animals, including poultry, cattle, pigs, and rodents. Salmonella can be found in contaminated water and food. People can become infected with Salmonella by eating contaminated water or food, or by touching contaminated surfaces and then touching their mouths. So once you get infected, symptoms of Salmonella infection such as diarrhea, fever, and abdominal cramps usually begins within 12 to 72 hours after the exposure. Most people recover from the Salmonella infection within a week without any treatment. But however, some people may develop more serious complications like dehydration or even reactive arthritis. There is no specific treatment for Salmonella infection. If people are severely dehydrated, they may need to be hospitalized to receive fluids through the IV and intravenous, which is a method of delivering fluids, medications, or nutrients directly into a vein. This allows the substances to be absorbed quickly into the bloodstream. In some cases, antibiotics may be prescribed to shorten the duration of illness. Among ways to prevent Salmonella infections are by washing your hands thoroughly with water and soap after using the bathroom, changing diapers, or before eating and preparing foods. Besides that, you need to wash fruits and vegetables thoroughly before eating them. Next, avoid eating raw or undercooked eggs, meats, or poultry, and remember, do not drink unpasteurized milk. That's all from me. Thank you.
Hi, my name is Richard Promdi, anak laki Ponchai, and my metric number are 22303083, and I'm going to be explaining about the clinical importance, but to be more specifically, I'm going to explain about the tendency of Salmonella enterica. Based on study from NIH from 2010 to 2021, Salmonella enterica has had over 11,269 reported cases with an average positive rate of 3.65%, including 1,614 hospitalization. This can be explained due to the trend. The first trend that I'm going to explain is the seasonal trend. It has the highest positive rate during the summer from May to October, accounting for over 77.96 positive rate. The reason for how the positive rate can get this high can be due to the preference for food during those times. For example, people during summer tend to prefer whole and raw food, which have been identified as one of the risk factors for getting infected by Salmonella. The other reason is due to the climate itself. Warm climate tend to be a much more suitable temperature for Salmonella to grow. The other trend are the age trend. The children and the older adults tend to be much more vulnerable to getting infected by Salmonella, especially the children from the age from 0 to 4 years old and the older adult with age more than 60 years old. The reason for this can be due to the immune system. For example, children tend to have a much more immature immune system as the immune system have yet to be developed, whereas older adults tend to exhibit a dysregulated immune response due to their aging. The aging can cause their immune response to be much more weaker and be more susceptible to getting infected by Salmonella.